Okay, so here we have the opiate test strip running. This is being dipped into a negative urine control solution, and you'll see in the central portion of this test strip the dye moving up towards the handle end, which is coded in this instance with MOP. This is a 300 nanogram high sensitivity opiate test membrane, and you'll see developing in the central flattened section of this test strip two lines, one nearest to the wavy end where the urine has been added to the sample test, and one nearest the handle end. That is the control line. The test line is the one nearest the dipped end, and this is a negative binding technology, so if there was any opiates at 300 nanogram concentration or above in this control solution, you would have no dye forming at the test line area and no line forming as a consequence. So you'd have in a positive opiate test just one control line showing with no test line at the read time, which for these tests is 5 to 10 minutes. And in this negative result you can see the clear two lines forming in the central section. Now, as we spoke in part one of this video, the opiate detection window after heroin has been used extends up to about 5 days maximum. Beyond that, the concentration in urine is simply too low to be detected, and the only way of detecting drug misuse at that stage would be to do a hair follicle analysis using gas chromatography and mass spectrometry. Now, that basically is a non-time dependent screen because the opiates would have been deposited in the hair follicle as it was being produced and grown the hair. So it's in there for as long as the hair is attached to that person for it to be analysed. Most drug screens using a hair follicle or hair sample would be cut off to around 3-month period of detection over which they'd look for any drug exposure. But the costs involved with doing those types of screening prohibit their widespread use in industry and workplace. So the majority of tests are going to be using a urine form of test strip, either a single strip or far more commonly where these test strips are embedded into a multi-panel either cup or dip test, so that a combination screen is being performed at any one time together with a heroin or opiate screen and other anything up to 10 or 12 common drug groups being screened simultaneously. If saliva is being used to detect heroin, then the window period drops to around a maximum of 3 days in most cases. And a lot of the membranes really will only detect opiates in the saliva for between 24 and 48 hours. The reason it's a shorter window is that the drug is detectable at much lower levels in saliva. Most membranes are working at around 40 nanograms in saliva, as opposed to the 300 or 2000 nanograms in urine. But also the metabolites aren't detectable in saliva. Obviously they're not being excreted through that mode. So these opiate metabolites that are being detected here, as well as the parent-based drugs, will only be detectable for a longer window in a urine-based product or screen. So that's why the majority of workplace and employment screening is still performed using urine as opposed to saliva testing. So hopefully these videos have answered the common questions surrounding how long opiates will be detectable in most drug tests performed using one of these one or two options. We will in a separate video discuss the 6-man heroin specific drug test and how that is being rolled out and its implications for workplace and healthcare screening.